Professor Keryn Williams, you moved from biochemistry and microbiology to looking at transplantation science and corneal grafts. A lot of people do go on a sort of circuitous route. I mean, your route was pretty circuitous, I think, we'd have to say. And tell us about what brought you to working with corneal grafts.
I took opportunities when they arose. Scientists can often work overseas, and I knew when I'd finished my training that I did want to work overseas. And I went to England, was there at the University of Oxford for almost 7 years. And that was a place where the Department of Surgery was just setting up a kidney transplant program. That was the first one in that area. And so I became interested in transplantation biology. And then when it was time to come home, as it were, I was offered a position in Adelaide by an ophthalmologist, so that's an eye doctor, who said he wanted a person to put some science into the art of corneal transplantation. Does everyone know what a cornea is? Do you know? It's the clear dome at the front of the eye. And if you think about it, if light needs to pass into the eye for us to be able to see it, it has to get all the way to the retina. And if there's any damage to the cornea through a disease or accident, and you end up with an opacity or a scar, then you really can't see. And it's pretty miserable being blind. It's not, you don't die of being blind, but it really does limit what you can do with your life. So it is possible to transplant a cornea, and that's always in the same way as it is for livers and hearts and kidneys. It's from someone who's died and whose family has given permission for the process. And when I first started working in this area, the ophthalmologist I was talking to said, look, there's this big myth that corneal transplants are always successful. I mean, it was in the textbooks. But the reality is, they told me, we know that they're not. So I work in a large group of people in the same way as everybody else here, a lot of collaborations. But we have a special collaboration. We set up a registry of corneal transplants across Australia. We've been running it now for 25 years, and we've got records of 23,000 Australians who've had a corneal graft. And we follow the outcomes of these transplants for as long as the patient lives or as long as the graft survives. And we have indeed shown what ophthalmologists knew from their personal practice, that a lot of corneal grafts fail. So some of our lab work is designed to try and fix that. But also, there's always a shortage of organs and tissues for transplantation. You know that. You've seen it on the news. When we looked, we found that corneas taken from people who died when they were in their 80s or 90s were just as good as corneas taken from people who'd perhaps sadly died in an accident in their 20s. And what that meant is that we could really expand the pool of corneas. Australia now is one of the few places in the world where there's no enormous waiting list for corneal transplantation. If you need a corneal graft, you'll get one quite quickly. No waiting required. So I think that has actually helped people. And I'm with our other speakers. It's really nice to do something that is practically useful.
And you're working with life and death, because people have to die to donate their corneas. And the sense of sight. There's a lot of poetry in what you're doing.
There's a huge literature and body of poetry on the importance of sight. I mean, I don't know that it's any more important than any of our other senses, but it is important, to be able to see. We are visual beings.
Well, next to me is Professor Brandon Wainwright, and you started with marsupials, amazingly
enough. I
enough.
thought you might mention that. I don't know why. So I'm a geneticist. So I work on genes, and so I started doing my PhD at the University of Adelaide on marsupials because, well, I won't bore you with the reason. They were very interesting from an evolutionary perspective. But during this project, I gained a lot of skills in how to isolate genes and how to manipulate them and to do various things with genetics. So I was sitting in Adelaide, and there was an ad in the back of a journal called Nature. It said, "Wanted person to work in human genetics, Imperial College, London, please apply." And I thought, oh, that seems pretty good. So I wrote, I got the job, and I went over there, and really unknown to myself, I ended up at the very beginning of the Human Genome Project. This was one of the 2 labs in the world, that were developing human genetics and the human genome. So kind of coming from marsupial genetics, I ended up being literally part of the first Human Genome Project, and I'm part of the committees and things that helped set it up. And then we became known as the gene hunters, the people who wade through DNA to find genes for disease. And then when we find those genes, try to develop drugs and diagnostics and other therapies, gene therapy and all kinds of things based on that genetics. So then I came back to Australia and picked up another gene, and we started working on a rare skin cancer, inherited skin cancer condition. And we found that gene, and it had a very strange name called hedgehog. So geneticists give genetic pathways, so like little switches in our cells. We've got hundreds of thousands of them. They often give them strange names, and this one's called the hedgehog pathway.
I thought you would have chosen a marsupial.
Well, there is, I won't. There is, look, there's a part of this pathway in another organism that's called echidna hedgehog and tiki-winkle hedgehog, and you're right. We won't go there. But, so the gene we found was the gene that's switched on, which causes the most common form of cancer, and that's skin cancer. And subsequently, it's called basal cell carcinoma of the skin. Hundreds of thousands of Australians get basal cell carcinoma of the skin. Consequently, not everybody, most people have it treated successfully. A reasonable proportion of people are disfigured by it. It's a very serious health problem, and some people die from the disease. So as a consequence of this discovery, a drug was developed which can now be used to treat basal cell carcinoma. And the thing we learned about genes along the way is that the gene which causes this most common form of cancer, when it's also switched on when it should be turned off, in other parts of our body, in children, causes a unfortunately all too common brain tumour called medulloblastoma. And in our lungs, it causes about 30% of all lung cancer. It causes virtually all tumours of our gastrointestinal tract from our mouth all the way down to the bottom bit here, apart from the colon. It causes a decent proportion of pancreatic cancer. And so the thing that we learned, as well as just finding out things about skin cancer and how to treat it, was that when we have cancer and common cancer, the body uses, things go wrong in a very, very defined way. And often by studying one cancer and thinking of therapies, we find a lot about other cancers as well. So this particular project was and still is a collaboration with Dr. Pratinder Kaur. Pratinder is a scientist at the Peter MacCallum Cancer Institute in Melbourne. She's an excellent scientist, a fellow of the NHMRC. And Pratinder is an expert on stem cells. And so we'd found the gene that causes skin cancer, and she was an expert in skin stem cells. So we thought, well, what do these genes do when they're not causing cancer? They cause the normal development of our body. So what we found in this project was that this pathway called the hedgehog pathway, if we can flick it on and off, and I won't tell you how we do that today, in the skin, we can turn a large proportion of the skin into stem cells, which we can then use, hopefully, then to treat people with burns. So starting with a simple, a rare condition, we found a gene, which turns out to be the gene that causes most common cancer, and then also it plays a role in how our tissues repair themselves as well. So that's how I went from marsupials to here. Yeah, sorry.
I just wanted to ask a question. I thought that too much sun caused skin cancer and too much smoking caused lung cancer. Now you're telling me it's genes.
Yep, that's exactly right. We're both right. Because everything that we are is a combination of the genes we get and our environment. And depending on what we're looking at in terms of just our characteristics, we've either got a lot of environment and not much genes, or things are very genetic and not much environment. Cancer is caused by damage to our DNA. And so damage to our DNA, so keep out of the sun. Seriously.
And don't smoke.
Yeah, yes. And so every one of these things damages DNA, which is why they cause cancer. And it's also why cancer increases as we get older, because we start out, obviously, we have one cell and it grows into 2 and 4, etc. And as it grows, the 3 billion chemicals inside our cells, the so-called bases of DNA, have to copy themselves every day or so. As we get older, and it makes mistakes, and as we get older, the proofreading mechanism, the thing that detects the errors, falls apart. So as we get older, at the molecular level, we fall apart a bit, and so as we get older, we have a higher chance of getting cancer. So the combination, as the Minister says quite correctly, smoking, cancer, no question about it.
All right.
It kills the genes.
Well, talking about cancer, Professor Glenn Marshall, for many people, children with cancer would be the most upsetting kinds of patients to have. And I imagine the most rewarding when you have something new to offer them as well, having patients with cancer. How do you negotiate the highs and lows of what must be very demanding work?
With great difficulty, Ramona. Our work at the moment is inspired by a number of things. I'm a clinician and a scientist, and I have no trouble getting out of bed in the morning because of 2 things: the children and their parents. It's absolutely amazing to me and truly inspiring watching parents do anything to make these diseases go away. And unfortunately in Australia every year over 600 children are diagnosed with cancer, and roughly about 150 of those kids are not going to make it. So we won an award recently, and the MC asked me, you're now getting an 80% cure rate for leukemia. Isn't this a wonderful result? And I said, absolutely not. From our point of view, it has to be 100%. And our patients live to 80 when they're cured at 5. And so the damage to the family, to the community at large is absolutely enormous. I get invited to 21sts and weddings and christenings and things now, which is really, a great part of my work. So that's the positive part. And the science is really come about through collaboration and colleagues. I'm part of a really large team of people, and was very lucky in the early part of my career to become very close friends and professional colleagues with 2 other scientists, Michelle Haber and Murray Norris, who we just remain scientific partners forever. And so we have a kind of a mantra in what we do, which is where we're making observations in the clinic, taking it back into the lab, trying to understand the cause of the disease, and then inventing something that's either a diagnostic or a drug to take that into the clinic. And we've actually done that in a number of diseases. But I might add that a recent leukemia trial where we invented a diagnostic began 20 years ago, actually, with a casual sort of conversation. And the trial will hopefully be published in the next few months, but it involves 1,000 patients, 3 continents, hundreds of clinical and scientific staff, and years of NHMRC funding, I might add, for which we would not be able to do it. The other sort of slight tyranny, I suppose, in the world of rare diseases, which childhood cancer is that the pharmaceutical companies are driven by profit, which is absolutely fine. I don't have a problem with that. It's just that we feel we're advocating for children. So if we weren't doing our work, then there actually wouldn't be anyone working on the targets for treatment and the treatments themselves.
Thank you. And Professor Mark Febbraio, your pathway to research wasn't straightforward either. You came via research physiology and doing some teaching, and your work on the heat shock protein has implications for a very interesting and different set of diseases. Tell us about your pathway.
Well, actually, it started before that. I actually did journalism, believe it or not, to begin with. So I didn't do science at school, and I was a not very good athlete, and I collapsed in a Ironman triathlon in Japan. It was in July, and I collapsed from heat stress, and I'd finished my degree, and it was sort of a light bulb moment where I understood that I was a pretty hopeless athlete and I better do something else with my life, and I then went and did a PhD in basically studying the effect of heat stress on metabolism. And we used to take little bits of muscle from humans out of their leg after exercise, and I did a lot of biochemical analysis.
A lot of volunteers for that.
And the first thing we did, we used to get volunteers, yeah. The first thing we did was discover that muscle makes proteins that talk to other parts of the body, and we invented the term myokine. And that was pretty good because nobody believed us. So when there's no belief in science, you get invited to a lot of meetings all around the world so people can poke fun at you. And so that was fun, and we used to go for entertainment value. And then I discovered that muscle made this protein called heat shock protein 72, and the question was, well, okay, what does it do? So my career transitioned more out of exercise physiology and more into cell biology, and we could take use of the mouse genome, and so we can So what my lab does is we knock out genes or overexpress genes in rodents, and then we can study whether this is actually very important. And to cut a long story short, we were able to show through the grant that was funded that if you activate heat shock protein 72, you can actually ameliorate the symptoms of type 2 diabetes, and we now have a chemical activator of this protein. It's gone through proof of principle trials in humans, and it's in a multi-centre clinical trial, and I'm associated with a biotech company with which we have this drug. But serendipitously, I was having a beer, which you also shouldn't do at your age and probably don't do when you get older either, but with my colleague at Melbourne Uni, who I'd never worked with but was a very good friend of mine, and he worked on Duchenne muscular dystrophy. And I read a paper showing that if you block inflammation in the disease, you may help the disease. So it turned out that our thoughts were correct, but our mechanism was completely wrong. But nonetheless, we made some what I consider to be, really interesting observations that were published this year, and we now hope to get the drug, which is already in humans, into a trial for Duchenne muscular dystrophy, which is so exciting because that's a horrible disease, because a child is diagnosed when they're 2 or 3 years old, and then they're a time bomb till they're 30. And the parents know, and the child knows, and if we can get a cure for this, it'll be terrific.